Welcome. Hello, everyone. Thank you so much for joining us today. My name is Rashmi. I will be your moderator. A couple of topics of housekeeping. Please enter your questions into the chat, and we will do our best to address them during the session. Your microphones are all muted, so you will not be able to ask the questions verbally live. But please feel free to chat in the chat box that you see. Also, if we do not get to your questions during the session, we will definitely address them by email personally. It's my pleasure to introduce Dr. Cromwell. So Evan Cromwell is the president and CEO of Protein Fluidics. He's a successful entrepreneur and an innovative leader of instrument and assay development. He has 30 years of senior executive and technical training experience in research, engineering, bioassay development, production, and marketing. Before founding Protein Fluidics, he served as the director of research at Molecular Devices and was a leader of cross-op scientific team for the Danaher Life Science Group. He was founder and president of Blue Shift Biotechnologies, a high-content screening and analysis venture that was acquired later by Molecular Devices in 2008. He holds a bachelor's degree in chemistry from Caltech and earned his PhD in physical chemistry from UC Berkeley. Dr. Cromwell has authored over 40 publications in the fields of biology, physical chemistry, and instrumentation, and holds 16 patents. And also congratulations, Dr. Cromwell, for the new publication that came out yesterday. So without further ado, I give you Dr. Evan Cromwell.
For the introduction, Rashmi. And today, thank everyone for joining us the end of March. Very excited about telling you about our PUMA system, where you've been using it for organoid assays, spheroids, tumoroids, any three-dimensional cell structures that people are now using to do really elegant biological assays. I really want to start by acknowledging the collaborators and other people who have contributed to the data I'm going to be showing you. The team at Protein Fluidics and at Molecular Devices, specifically Oksana Serenko, who's done a great job using our system for imaging and coupling with high-content imaging. Marguerite and Matt Burrow at Tulane, who provided the patient-derived tumoroids and cell lines for us. And the teams at Griner and Promega have been assisting us with different technologies for accelerating the assays in our platform. So today, I think really three sections. First, talk about 3D cell models and their use in the industry, a little bit of perspective on them, and then the second would be on our PUMA system. What's the technology behind it? How is it used for doing assays with organoids, spheroids, tumoroids? And then the third section is really getting into data from our users, what are the applications they're doing, and where does our system really accelerate your assays and automate the technology assays that you want to do on your benchtop. So I look back at the history of 3D cell models. It really goes back to the 50s, when people started wanting to refine, reduce, replace, refine animal testing and move to cell models. In the 70s, they started making HeLa spheroids. Some of the data I'm going to show you today is HeLa spheroids, which is now from models developed 50 years ago. Another big breakthrough came in the 80s when people started using the stem cell derived lines. And this really opened up the doors to doing humanized models. And then with the advent of induced pluripotent stem cells, 2005, 2006, that really opened the doors farther because now people could use these models and create organ-specific cell lines that were not derived from human fetuses, et cetera. And so politically it became much more tenable. And with that, then the US government got involved with the Tox 21 program, tissue chip program, and now in 2021, you have some really elegant organ-on-chip, multi-organ models that people are using to really recapitulate the human body. 3D cell models, you can look at them, where the big press comes. It comes from high throughput screening. There are people doing 1536 well organoids or spheroid screening, and the low throughput analysis, these multi-organ on a chip devices. And if you look at the differences between them, you're going from a huge number of samples being analyzed and number of data points acquired to small number of samples, but very complex data. And in addition, the models, when you're doing the high throughput, you necessarily have to go to simplicity. So single cell line spheroids, whereas in the low throughput, you can add a lot of complexity into vascularization, organ-organ communication, etc. But where do we fit? It's really in the middle, where you want an open platform, you have an established cell model, and if you want to automate the processes, you want to improve the data that you're collecting, get superior results from those models. Another way to look at this is really the drug pipeline, where you start from the target ID, going all the way over to clinical development, clinical candidates. And you can look at each one of these buckets, and somewhere in there, people are starting to use 3D cell models. Where we think we fit best is really in the early assay development part. Not really high throughput, I would say we're low to medium throughput, but doing lead optimization, doing multifunctional profiling, and then moving into the preclinical area where you can use our device to really help guide therapeutics. Now, there are a lot of lab-on-a-chip technologies out there, and that's all nothing the technology that's been developed over the past 20, 30 years or so. But most of these devices are going to be fairly complex. You have to do a lot of connections to them. You have external pumping. They're made out of PDMS or other materials, which are a little harder to put together, a lot of clamping involved. But they do give you the small volume, smaller footprint, fit on the benchtop. One of the goals is to reduce or replace expensive automation, but the chips themselves can be very expensive. And then often these are single purpose. So you have a very complex device, but it's really geared towards doing a single type of measurement. So our solution is really to create an open lab-on-a-chip platform, if you will. We want to make it as easy to use as possible, fit right into the workflow of the common laboratory, and then work with a variety of sample types. So any type of 3D cell model that you can create, you can put into our Flowchip, then you can automate processes with those from 100 microns up to around 1 millimeter or so. And we automate the transfers of media, we automate staining, we automate a number of these processes that are done manually right now by people. But the advantages we provide is you can do this without disrupting or losing your sample, doing it in an incubator so you don't have to take it out, you don't have to disrupt or perturb the biology, if you will. And then it's really designed, we don't do analysis in our system, but we provide a system that is a platform that is compatible with all your downstream analysis. So the workflow then is you have your flow chips, and I'll describe these in the next section, where you load your reagents and your spheroids or organoids into those. Those are then placed in our PUMA system. PUMA is for pneumatic microfluidic assay, that's the acronym. In the incubator environment, the system stabilizes at 37 degrees. Pneumatic is because we use pneumatic pressure differentials to move fluids from one well to another. And a consequence of that is we are constantly pulling in air from the outside the system to create pressures. So we're constantly recirculating the air in the incubator environment to the media over the chip. So you're maintaining 5% CO2 at your media. And then the system is all controlled by a microprocessor, which contains preloaded protocols. Those protocols detail every step in your assay. So all you have to do is select the protocol you want to run, incubation, staining, metabolite sampling, press play, and then let the system do its job. And when it's done, pull the plate out, it's ready for analysis. So let me talk a little bit about the details of the flow chip. As I mentioned, it's designed to fit into your laboratory flow, your laboratory automation. And the first thing is to make it compatible with the multi-well standard. So this is a 384 well spacing. Looks sort of like a 384 well plate, but it's a series of flow chips. And then each flow chip has 8 lanes, and each lane is one test. And so in the lane, you have this center well where you place your spheroid or your organoid. You can see an example of a transmitted light image shown here. And then there are 8 wells connected to that. So you can transfer fluid in and out of here from 8 adjacent reservoirs. And if you look at cross sections, about a 20 microliter volume here. The organoid is kept in this protected sample chamber, which I'll show a little more detail next slide. And then flows can come in and out of that sample chamber from any one of these wells. So you have a very an open platform where you can multiplex different fluid movements and you can go back and forth to the wells. A little video here shows how you would dispense your organoid into the well. It nestles down into the sample chamber. And then as you move fluids in and out, that little chamber within a chamber doesn't drain. So it maintains fluid around your structure. You don't have flows going directly by your structure, so you aren't creating shear forces, you aren't pushing it down another microfluidic channel. So it really keeps this nice and healthy and happy in its little protected chamber. And then when you want to do automate steps, doing fluid transfers, here we're showing just sort of the standard type of things our customers would do, which is changing the media, doing a media exchange, where you remove the old media, add in new media, incubate for a period of time. Then you could do a supernatant sampling, where you transfer in fresh media, incubate for a prescribed time, and then transfer that back where it can be sampled later for analyte measurements. You can do staining. If you need to do a wash after staining, you can remove the stain solution, add in a wash, and now it's all ready to cut onto your imaging system. So you can imagine, the different types of movements you can do and the type of assay protocols you can build up with this device. And then in the idea of keeping it as simple and easy to use as possible, all those protocols are preloaded into the system, and you can just select whichever type of protocol you want to run, whether it be an overnight, 2-day incubation, whether it be staining, etc. So you've really identified, the assay protocol steps that we sort of add the most value to, do the best job, and a lot of those, as you can imagine, relate to just doing simple transfers. But doing time course sampling, I think, is really key, where you can pre-describe, how often or when you want to remove sample or look at metabolite secretion. I'll show you an example of that. Or if you want to do sequential drug addition, you can increase the concentration of the drug. You can do multiplexing of drugs and add that at a pre-described time. Because you're doing this in the incubator, you can in situ lyse your cells. And by that, I mean your cells are in the incubator environment, and then you remove the media and you come in with a lysing solution, all without having to take it out or perturb, sensitive metabolism in your cells. And then from the immunofluorescence staining, there's a number of steps involved in there, whether it be your primary antibodies, blocking, washing, secondary antibodies, more washing. And so those number of steps, it could be 8 or 9 steps, and we can set those up on our system, do that all automatically. Then the type of assay results, and I'll show the 3 examples of this in the next section. We can look at secretion, viability, metabolomic profiling. I really haven't mentioned it, but our Flowchip has a thin COC bottom, 140 micron thick COC bottom, which is compatible with very high resolution imaging. You can go up to 60x, even 100x objective if you want with our system. And I think that's fairly unique among microfluidic devices. And so it really makes our system compatible with putting onto a high content imaging system and collecting the data and then doing subsequent profiling of your organoids. Okay, so from applications, there will really be 3 specific ones. First is just doing single cell spheroids. These are HeLa, HepG2 spheroids that are formed, showing cell viability, phenotypic profiling, and sort of what we call multifunctional profiling. The next example will be tumoroids, and these are tumoroids which are formed from patient-derived cancer cells from a triple-negative breast cancer patient. And lastly, some examples of organoids, more complex structures that you can use our chip to treat and to image to get really high resolution, high content data from. So for cancer spheroid profiling, in this case, we would form the spheres. We actually use a technique called nanoshuttle, and I'll show that in the next slide. Ran the assays in Puma system, and then after the overnight incubation is done, you can add cell stains to do viability measurements. You can see phenotypic changes by the differences in the organoid shapes, and then sample supernatants to look for secreted factors. And then, so this is all 3 sort of endpoint reads you can get from a single spheroid. So the magnetically printed spheroids. And this is doing magnetic 3D bioprinting from Griner. In this technology, you can coat your cells with small magnetic nanoparticles and use a magnet to bring them together in your low attachment plate, the cell repellent plate. Then after you remove the magnet, then they'll form naturally into a spheroid. We can then use that magnetic property to put a little magnet underneath our plate and use that to position and hold the spheroids in the center of the plate it improves your imaging. If you're having any worried about loss of the spheroids, then it holds them in the middle and down at the base of the plate. The nanocell itself doesn't interfere with your biology or your imaging. This is showing really high resolution, this 20x water immersion image of a HeLa spheroid that's been formed using the magnetic 3D bioprinting and then placed in our system and stained with nucleoapoptotic stain, a micron stain, and actually Hurst nuclear stain. You can see very high resolution images of the cells and be able to look at cell cycle mitosis, for example. And with that, and then you mentioned these were taken with the ImageXpress microconfocal system. And then using the MetaXpress analysis package, you can then, with each spheroid, identify single cells and classify those cells, whether they're alive, they're dead, apoptotic, etc. So your typical high content imaging analysis is all feasible with imaging on our plate because of the optical clarity of the bottom. So here you can just see with the different types of treatments, you can see the requisite killing of the cells, killing the spheroids, or arresting them in apoptosis, for example, by mitomycin C. And then you can do concentration response of that. Here we're doing a cytosporin treatment, and you can see the live dead curves here, giving you IC50s for cell viability. And then for each one of these spheres, we sampled a supernatant and did a VEGF analysis by ELISA. With the HeLa cells, we didn't see any measurable secretion of VEGF, but for the HepG2, we saw a very nice secretion, and going from to 3 nanogram per ml down to negligible for the high treated. So you now get the concentration response of VEGF secretion as well as the concentration response of cell viability, all on a spheroid by spheroid basis. And so that I think makes really unique about our systems. Now you can think about looking at heterogeneity of your organoid models, your spheroid model, because you can do single spheroid interrogations. So taking that into a more relevant, more important cell line, this is patient-derived tumoroids. Again, this came from our collaborators at Tulane University, Matt Burrows' lab. And these were a cell line that was extracted or derived from a patient with metaplastic breast cancer, a triple-negative breast cancer subtype. And importantly, this patient the tumor resisted therapy to chemotherapy, specifically paclitaxel. Now, Matt's lab works with xenografts, he does the organoids, and he also does spheres. In this case, we're taking the cell line that was derived from this tumor and then plating those cells into ultra-low attachment plates and then allowing that to form a sphere from these, we call them tumoroids. And then for using the magnetic properties to plate these, we then added a little of this nanoshuttle in there, and that coated the spheres, the tumoroids, magnetically. And so now with that, we can then also use the magnets to pull them down the bottom. Now with the big spheroids, it's not as critical, but with these smaller tumoroids, these are maybe 150 microns, in diameter. Having them centered in the well is going to be very important if you want to get good imaging analysis from these. Without that, they tend to go to the sides of the well. They like to go to the low energy places. You also see here just to transmit light. They're fairly translucent, fairly healthy. You don't see any interference from the magnetic nanoparticles. So what have we done with these? We've done, again, this multifunctional profiling. We did viability assessment, metabolite analysis, and immunofluorescence staining. So for the viability, similar to the spheroids, we place the tumoroids into our Flowchips, incubate them overnight with a variety of compounds, and then for the staining, because the stains are not stable overnight at 37 degrees, we would remove the plate, add the stains in fresh, and then do the staining in a separate protocol step. But the easy addition and removal of a plate to our Puma system facilitates those type of steps. And so once the staining is complete, you can sample your supernatants and do the imaging. So here's some representative images of the tumoroids treated with romidepsin on top and trametinib on the bottom. This data is Marguerite has submitted for a manuscript, which should be coming out hopefully soon. And then you can see, again, using the same type of high content analysis as I showed for the spheroids. Here you can classify the cells, individual cells in the tumoroid as live or dead, and see concentration responses. And significantly here with romidepsin and trametinib, they're in the low nanomolar range, which is consistent with the patient response and also consistent with other cell models response to these compounds. But paclitaxel is factor 100 to 1,000 higher in EC50 value for its treatment. So the patient was resistant to paclitaxel treatment or Taxol treatment, and we were able to recapitulate that in the tumoroid model from this patient in our Flowchip. The second sort of analysis we did on these tumoroids was Metabolite, and this is something new that we've been working with and working with our friends at Cromwell. It's an interesting setup because with the ability to sample and do a time course, you can now think about looking at changes in metabolism, metabolite dynamics from single tumoroids or single spheroids. In this case, we would sample supernatant, transfer back and forth at different time points, and then from each one of those samples, we have 20 microliters, but you can do high dilution because of the sensitivity of these assays. So now you can think about doing 4 or 5 different analytes from your sample. So this is each measurement will be 5 analytes from a single tumoroid. And they're using their glow kits here, and they're recording the luminescence on their GlowMax system. So to help you visualize what that means in our Flowchip, you would preload the media or medium compound into the wells, and then at pre-described times, you would just transfer media in, let the tumoroid incubate for that amount of time, and then transfer it back, and then repeat that. So after doing that for 5 repeats, including the initial removal of the media, you now have 5 samples here. So we have one where we use it as our control without cells, and we had the 4-hour, 8-hour, 12-hour, and 16-hour sample time. If you then measure, in this case, they're showing the dynamics of lactate secretion with paclitaxel treatment and romidepsin treatment. The scatter tumoroid to tumoroid the variability is pretty high, but this is the average of 4 different tumoroids together. And with that, you can see that with Taxol, you actually have an increase in lactate secretion, increase in metabolism at low concentrations and lower time points. With romidepsin, you're not seeing this change in your metabolite dynamics. So it's interesting, and it's actually confirmed in the literature on this paper 2019, where they're doing pretty much the same experiment, and they were seeing that the lactate production increased with Taxol treatment for a Taxol-resistant breast cancer cell line. Some of the differences in the experiments, though, in this case, they're using a 96-well plate, 20,000 cells per well, and sampling the media, sampling 10 microliters of media from 200 microliters over different time points. And so they're really looking at integration of the lactate in the well, whereas what we're doing is we're doing actual time splices from single tumoroid. So reducing the number of cells by a factor of 10, increasing the time point measurements, and increasing how much information you're getting from that. So slightly different, but again, seeing the same type of result from Taxol-resistant cell lines and seeing an increase in the metabolism at the low concentration and lower time points. And then because we have this tumoroids in our plate, after we do metabolite sampling, we can add viability stains and then look at the change in viability with the cells. After the 16-hour treatment, the cells were still viable, so you can see the core of the tumoroid here still has a calcium positive staining, but you can definitely see an effect, and the IC50 of this was similar to what we saw before from the viability experiments. Okay, so last for this is really immunofluorescence staining, and as I mentioned previously, with the 8 different wells, it really is set up for doing more complex type of protocols where you have multiple transfers that you have to do. And the challenge when doing these with single tumoroids in the multi-well plate is you're doing manually, you're often restricted to only removing half the fluid because you're concerned about pipetting up the tumoroid, or if you get the pipette tip down, you could damage the tumoroid. So it makes it really difficult for doing sort of manual immunofluorescence staining, and we automate that process. So this is, again, 2 tumoroids that were, one was a control. This is after overnight incubation, and stained with E-cadherin in green, it kind of shows up yellow, and then CD44 as red and positive, and some DNA stain in the background. Again, these were done with image trust microconfocal, done by Oksana Serenko, using a 20x water immersion objective. These particular ones is a confocal stack that we're showing you maximum projection image of the tumoroids. And then you can do this for different compounds. And so again, the paclitaxel, romidepsin, trametinib. You can see the EC50s. Again, paclitaxel has a much higher EC50 and it's consistent with the taxol resistance of this patient. And these actually derive from the CD44 markers. So CD44 being a marker for live tumor cells, cancer stem-like cells. But the E-cadherin drops off really fast. So we're looking at how the E-cadherin response is, and that work is ongoing. So last, I want to show you some organoid assays. These are examples of lung organoids that were grown in Matrigel. And so this is, again, work done by Oksana Serenko using stem cell technologies, lung organoid compounds, and medias. I think it took 7 to 8 weeks to form these in Matrigel. But trying to image in Matrigel is difficult, too much distortion for getting really high resolution. Also treating them has its complications. So in this case, we can just dissolve away the Matrigel and then place those organoids into our flow chips. And so now you have the ability to do high resolution image, to do metabolism analysis of these, to do other secreted factors and look for effect on viability, even do look at cell surface markers, etc. Doing that all in the Puma system. So interestingly, for these, when we loaded them in our flow chip, you can look like they're sort of suspended in midair, and they are suspended in the Z. So some residual Matrigel is there and keeps them sort of in sort of 3D, and overnight incubation, you see them maintain their shape. So that's really a positive for these type of organoids that are more sensitive to the extracellular forces on them. In this case here, we're doing again a 10x objective, so in calcium M and Hirsch staining, and then going to a little higher resolution and doing a confocal stack. What you can see here is these are actually hollow. Very interesting, and allows you to get really nice confocal image stacks. With the spheroids or tumoroids, which are more compact, it's very hard to image the centers, but here you can see the ring of cells around, very nice projections and stacks. And then as you go through this Z stack, you can see these are really floating in 3 dimensions here. So we're very excited about this capability. Obviously, the pictures are really beautiful and stunning, but when it comes down to it, you want to run an assay, you want to look for data from these, and so you can do the same type of treatments here. Here we're showing a Bioprinted treatment for 48 hours, and then after that 48 hours, being able to do the imaging in an image trust microconfocal system using MetaExpress analysis for live and dead, and see the effect of compounds on these organoids. So with that, I'd like to sort of recap what I've been trying to show you during this webinar. We've really developed this Puma system as an open lab on the chip to facilitate organoid, spheroid, tumoroid assays on the benchtop. We're not, high throughput automation. We're not doing 1536 wells. We're not doing these complicated organ on a chip and doing organ-organ communication. But for the large majority of assays that people want to run and interrogations that people want to do with these 3D cell models, I think our system fits in very well. We have unique capability to protect your spheroid, your tumoroid during the processes. The system allows very high resolution imaging, so you can do this multifunctional profiling. You can do high count analysis, you can do metabolite analysis, you plate beater analysis. We're working on some type of live cell kinase assays with Promega and others. So there's a wide variety of assays you can do with this. And emphasizing the fact that you can now do this on a single tumoroid, single spheroid, single organoid basis. And then you can look at heterogeneity of your organoids and compare organoid to organoid response. So with that, I'd like to thank you for attending. I can be reached here at my email, ecromwell@proteinfluidics.com. You're welcome to go to our website, www.proteinfluidics.com. And I think we have time for questions. So Rashmi?
Thank you so much, Evan, for a nice presentation. So a couple of questions that did come. So one of the questions from Christine was, does the PUMA system actually capture images, or do you need to move to a microscope?
So the PUMA system does not have imaging capabilities. And so you would remove the plate and put it into your imaging system, put it onto a microscope, etc. The flow chips are designed if you're doing any work with multi-well plates. It's like a thin bottom COC multi-well plate, but we purposely didn't add in the complexity of imaging to our device because we don't want to create another high-content imager. We want to create an organoid automation device.
So another question from Christine was, when you do a whole plate, can you do different stains in different rows?
Yes, you can. So each lane on the flow chip, each lane on here is really an independent test. And so you can put different stains into the wells. You can look at different outputs if you want. You can do different immunofluorescence stains in them, with the caveat that the incubation times will be the same for each lane. So each lane you would transfer a different stain, different antibody, but it would reside in the sample well for the same period of time. So if you want to do 2-hour incubation, that was sufficient for your different antibody stains, and that can be done. But each lane is an independent test. It's going to be different antibodies, different compounds, different stains, etc.
So a question from Hector is, what is the maximum size a spheroid inside the chamber can achieve?
So the maximum size is around 1 millimeter. The diameter of chamber is about 1.2 millimeters. A 1 millimeter sphere will fit down there, and then the height's about 1 millimeter. And then the minimum size we've done 50 to 100 micron tumoroids.
Okay. The next question from Yong is, can your 3D system solve the challenge of blood-based oxygen delivery?
I'm not quite sure about blood-based oxygen delivery. If you're referring to hypoxia, it can. So the system itself is sitting in a chamber. So because the system is pulling in the outside air, if this is an hypoxia chamber, the oxygen levels at your media, oxygen levels that your cells are going to see will be the same as outside. Now we're also working on a system where we can control the environment without being an incubator. That's sort of in the early stages right now. But essentially when we seal this pneumatically, we're controlling the environment above it, and whatever is in your incubator here or in your hypoxic chamber, that will be replicated at your media. So you can do low oxygen hypoxic experiments with our system.
Those are the questions that came in so far. If anyone has any further questions, we'll just stay online for a couple of minutes more, and even after the session is ended, so we can address your questions. Or else you could also email us through ecromwell@proteinfluidics to Evan directly, or you could also leave us a comment on our website. We'd be happy to reach out back to you. Also, I just want to let everyone know you will receive an email with the recording of the session, so you could review it, and all this information will be there, and our email addresses for you to reach back to us. Thank you so much, everybody, for your time, and thank you, Evan, for the presentation, and hope you all have a wonderful rest of the week. Thank you.
Bye, everybody.